Skeletal muscles move bones by contracting. During contraction, the muscle shortens and thickens. When the muscle relaxes, it returns to its normal shape and length. Muscles contain many bundles. Within each bundle are many long muscle cells. These cells are called muscle fibers. Unlike most body cells, muscle fibers have many nuclei. Each muscle fiber has a pattern of light and dark bands or striations. The thin dark lines in the middle of the light bands are called Z-lines. A pattern of bands and Z-lines runs the length of each muscle fiber. The muscle fiber is made up of myofibrils, which in turn are made up of muscle filaments. It is these filaments within the muscle fiber that give the cell its banded appearance. There are two types of filaments within a myofibril, thick filaments and thin filaments. The Z-lines are actually disc-like sheets of protein to which the thin filaments are attached. The segments of the myofibril between two Z-lines is called a sarcomere. The thin filaments are constructed of a protein called actin. The thick filaments are bundles of protein molecules called myosin. Each myosin molecule has a tail and a head region. The tails of the myosin molecules clump together, while the heads protrude outward. Some of the myosin heads are attached to actin molecules, forming cross bridges. The myosin molecules are oriented with their heads toward the ends of the thick filament. As the muscle begins to contract, it shortens and thickens. Now think about the sarcomere. When the muscle is relaxed, the free ends of the thin filaments are far apart. The Z-lines are also far apart. As the muscle contracts, the thin filaments are pulled toward the middle of the sarcomere until the thin filaments almost completely overlap the thick filaments. The Z-lines are pulled together, shortening the sarcomere. As a result, the muscle shortens. When the muscle relaxes, the sarcomere lengthens again.